change. So, but first I would like to introduce Petra Dillman, who is the president of the World Autism Organization, and she will share with us some information about the World Autism Organization. So, Petra.
Good evening or good day, everybody, depending on where in the world you're listening or you're looking from. I'm Petra Dillman. I'm a parent of Michael, who's 35 on Friday, and I've been involved in autism for most of his life. And I'd like to introduce the World Autism Organization. That was formed in 1998 and formally constituted and had its first president, so it's like 25 years old last year. And it stemmed, or it sprouted out of Autism Europe, because there were many countries outside Europe who were requesting help and programs and projects, so that the Autism Europe group decided that it might be better to have a World Autism Organization to be able to handle all those kind of requests. So then from 1998, we've had conferences nearly every 4 years, starting in Melbourne, then in South Africa, in Mexico, in Kuwait, in Houston, and then came COVID. So nowadays, with COVID teaching us all to be more online, we are planning to do conferences online. In the meantime, the World Autism Organization, in collaboration with the Autism Media in Turkey, are endorsing conferences that are being held in the Eastern European countries, mostly spreading to more Middle Asia. I've actually looked on the map the other day, and there are just many, many countries in that area, just like in the rest of the world. The World Autism Organization is made up of volunteers, and they're coming from England, from Italy, from Kuwait, from Germany, from South Africa, myself from Namibia, from Mexico, the United States, and some other countries as well. We've got many members, but not everybody is on the Council of Administration or the executive, and we are grateful for everybody's voluntary help. Our other things that we're doing, we've started last year with like a parent support program, which is also online, whereby we record various topics that parents keep on asking about, and they're going to be, or some of them are already online in a separate YouTube channel. And then thank you for ARI for allowing us to host with them every now and then. We also put all those links to the ARI webinars on our site as well. So part of our function is also to network and bring together organizations and people to teach them to help themselves. The need is great. The questions and the request come, they're all the same, that it's parents that are looking for better opportunities for their children. They don't have services in their country. And this is so much, and funding is so difficult to get, that it's better to give practical advice. And again, thank you to our volunteers on the Council of Administration and other friends within the World Autism Organizations for giving their time of hosting and recording these webinars to be able to help people all over the world. Thank you very much. And if you'd like to see our websites, it's www.worldautismorganization.com, and the organization is spelled with an S, not a Z. Thank you very much.
Oh, thank you, Petra. Now let me just say a few words about the Autism Research Institute, or ARI, or ARI. ARI was founded by pioneering researcher and parent advocate Bernard Remland in 1967, so that's 57 years ago. And over the years, we have monitor research, support research, and also conduct research. More specifically, we fund research studies sponsor an international support group network with over 260 member groups, as well as a science network. We sponsor think tank meetings, publish original articles and books. We also produce videos, webinars, and a lot more. Actually, recently we created an online app to provide parents and professionals a way to assist in determining how to treat sleep disturbances in autism. And by completing this checklist, the user is given links to published research to help understand and treat specific sleep problems. So this is rather a new innovation in the field. And so you could visit the website, you either go from our website, autism.org, or simply go to www.sleepdisturbances.org. So it's my pleasure to introduce Dr. Antonio Persico. His main focus of study is the interaction of genetics, epigenetics, and neurobiology. This work involves clustering autistic patients based on their biology, identifying biomarkers, and then predicting appropriate therapies, as well as developing new pharmacological, behavioral, and occupational interventions. Currently, Dr. Persico is at the University of Modena and Reggio Emilia. So, Tony, you could correct me. So, please welcome a longtime colleague of mine, Antonio Persico.
Thank you. Can you hear me?
Yep.
Yes.
Okay, perfect. Thank you very much, Steve. I'd like to thank the Autism Research Institute and the World Autism Organization for inviting me to hold this webinar. And let me start off by expressing, though, my profound grief and my greetings to all people from Baltimore and from Maryland. Baltimore is really my hometown in the US. I spent there some of the best years of my life. I've been over Key Bridge for dozens of times, and I really would like to express all my sorrow for what happened 2 days ago, and all my best wishes to Baltimore and to Maryland for getting quickly back up on their feet and putting back things the way they should be. What I will try to do today is to provide you with a little bit of a different approach to biomarker research. We tend to view biomarker research as a little bit of a list of items that we would like to be telling us which patient will be autistic and which may not, or who would that we would like to tell us essentially what we cannot fully grasp clinically. Now, biomarkers can do a lot more for us. Biomarkers can really tell us what's behind the origin of a disorder, provided that we make the effort to consider the complexity of the pathophysiology of complex disorders and to merge information from different approaches. So this is what I will be trying to do today, limiting, of course, myself to the time I have, and therefore limiting to some paradigms that I hope will be useful to you. In general, as I was saying, biomarker research focused on the identification of panels of biomarkers that we would like to help clinicians in providing a more appropriate diagnosing, decreasing the risk of false negatives and false positives, which is always present in clinical practice. We would like to have pre-symptomatic risk estimates, possibly even at birth, that would help us predict which children should perhaps receive very early behavioral interventions, even preventive interventions, because they are at greater risk of developing ASD as they grow. Predict developmental trajectories. We all know that there are children that improve a lot but there are also children that receive the best possible treatments, intensive treatments, and that don't improve as much. So we would like to know which children should receive which treatment, because not all treatments are equivalent by far, and not all children respond equally well to all types of treatments. To design personalized pharmacological and non-pharmacological interventions, and ultimately, and this is really what triggers my interest the most, to begin understanding what's behind the complexity of this disorder that we call autism spectrum disorder, and whether we're really putting together apples and oranges. We're really putting together patients that have very different origins and explanations into something that merely shares a superficial clinical presentation. Now, there are multiple levels of analysis that one can apply when attempting to perform biomarker searches. There is genetics, epigenetics, there is transcriptomics, proteomics, brain imaging, metabolomics, and going all the way up to the more complex and closer to the behavioral phenotype, eye tracking, electrophysiology, movement kinetics. And of course, during the first decades of research, each group, being expert in essentially one approach, tended to really focus on one level of interest, and was hoping to find at that level an explanation that was sufficient. What I will try to demonstrate today is that really we can only grasp some of the complexity that's behind autism if we really do the effort to put information together from multiple levels of analysis. Starting from the genomics, which is really the closest level to the biology of a disorder. But let's remember that in a brain disorder, genetics is also the farthest level from the complexity of behavior. Now, if today we were to apply all the possible genetic analysis that we can perform in a patient with ASD, we would probably come up with about an answer in terms of what we call a causal gene, causal mutation, a causal deletion or duplication, or a probably pathogenic anomaly in approximately 1 patient out of 5. And if we are dealing with more severe cases, with also intellectual disability, nonverbal, etc., it would be probably in about 50%. Now, while this is extremely interesting and valuable, it is far from what we were saying only 15 years ago. I myself was saying that autism is really genetic, and maybe not 15 years ago, but 20 years ago, when I started off, 1997, working in genetics of autism, we were saying that 3 or 4 major genes would explain the vast majority of autism cases. I'm sure that many of you remember those days. Now, the more we work in this field, the more we find that that's not the case. So while these percentages are very relevant, they still are far from what we were expecting. And what we find is that in reality, it's relatively small. We've seen the number of patients for which we can identify a rare variant. I don't know if you can see my cursor, but I will describe the red arrow would be a rare variant of high effect. So one of those that has almost certain penetrance, which is probably almost alone by itself necessary and sufficient to move you from the white part of this graph, of this curve, to the yellow part. Other patients will have what we call incomplete variants with incomplete penetrance. So they do increase the risk in a sizable way. They are rare variants, but they are not sufficient by themselves to produce autism. And in fact, sometimes they are inherited from parents who may either be entirely non-expressing the phenotype or may have some minor traits. And still, there are many patients who end up being like the bottom line here, just many small black tracts, which are common variants. Common variants like those variants that you and I carry, many of these, each one contributing maybe 1-2% of the variance toward satisfying the criteria for a disease. So clearly, there is an interaction between rare and common variants. Clearly, it is relatively rare, not entirely rare, but let's say unusual, that a patient will be entirely explained by a single genetic variant. And when I started off, and I think many of you will remember, autism genes were just very few and scattered in these graphs here. They were scattered on very few chromosomes. Nowadays, look how many genes are so-called autism genes, meaning that if they're mutated, deleted, or duplicated, they will likely lead to a neurodevelopmental phenotype. And in fact, if we analyze, like Thomas Boucheron's group did in 2021, if we analyze the results of studies that reflect which genes are known with high confidence, if mutated, to produce neurodevelopmental disorder phenotypes, and which genes are candidates. So this would be SFARI code number 2, 3, and S, for example. You will notice that high confidence is 1,586. It's an enormous number. Candidate genes are over 6,000. And considering that using this technology, which I believe I remember was exome sequencing, you can exclude not all 33,000. You don't analyze 33,000, but you get data on a lower number. You can exclude 11,000. So essentially what this is saying is that aside from those relatively rare cases where a single gene really explains it all, but mostly the genetic of autism is beginning in some way to look like the genetics of epilepsy, to look like the genetics even, I would say, of type 2 diabetes, where you have strong familiarity, but the underlying genetics is really, really complex. To try and tackle this complexity, we can already start using strategy like functional analysis. So rather than searching for the autism gene, we may ask ourselves, those so-called autism genes, of course, they encode proteins. What are the functions of those proteins? And essentially, what is a recurrent finding in the literature is really that there are two large functional families of genes that are neuronal communication genes and that are expression regulation genes, which are really consistently over different studies encountered as being the main functional categories. Interestingly, in this beautiful work that we gave a little contribution in terms of some families by Satterstrom and colleagues 2020, these expression regulation genes tend to be mostly expressed prenatally, whereas neuronal communication genes start being expressed prenatally, but they are also heavily expressed in the first few years of life. So this is one interesting way that we can begin tackling the complexity. Rather than thinking of autism genes, think of autism-related gene pathways and functions. And as I said, other studies, also on large numbers of patients, had found similar results. You can find here, for example, this work by the Rubes et al. Transcriptional dysregulation is really a synonym of expression regulation genes. You may find synaptic transmission and neurodegeneration, which is really also a synonym of neuronal communication genes. In some studies like this one, you also find a third category, which is immune genes. And immune genes here, TGF-beta pathway, are something that we shall go back to because of the complex interrelationship between neurodevelopment and immunity, which we'll be just briefly touching by. In two studies that we ran on our sample, 329 autistic children of Italian descent, and we also looked at 98 autistic children and adolescents from a Tunisian sample. We pretty much replicated the results that we have shown before, namely that we have about 23% of positives, here you can see, and that pathogenic and probably pathogenic CNVs, these are CGH results, and that essentially in both samples, you really have neuronal connection pathways that are mainly involved. So we don't find strong evidence of major differences in the genetic contributions present in different socioeconomic and ethnic contexts. There are, however, additional layers of complexity that we must take into account. First of all, we speak about autism genes. Is there such a thing as an autism gene? The response is no. We should call them neurodevelopmental genes, essentially because the same gene can be mutated, deleted, or duplicated, and it may yield autism in one patient, it may yield intellectual disability in another patient, it may result in both, it may result even in additional, in other neurodevelopmental disorders. So essentially, the human genome does not respect the DSM-5. In addition, we work a lot on Phelan-McDermid syndrome. We have a sample of over 80 now patients with this rare disorder, and it's common in our clinic to see patients that have almost identical deletions in chromosome 22 in the long arm of chromosome 22, and who may be absolutely incredibly different in terms of severity, in terms of which systems and organs are involved. So clearly there is huge pleiotropy produced by even huge pleiotropy produced by similar genomic anomalies, even in genetic syndromes, which are supposed to be relatively homogeneous. Finally, imagine you have in front of yours, in your office, a child with Tourette syndrome. And imagine that child with Tourette syndrome is recruited for a genetic study. If that child with Tourette syndrome has an uncle with OCD, so if the genetics to Tourette passes through the OCD pathway, that child will have a GWAS result, which will be the one that you see in B, below here at the bottom right. But if that child has the identical clinical picture, but really has familiarity for ADHD and ASD, his GWAS results are going to be totally different. They're going to be panel A. And clinically, these are so close that you will put them in the same sample, essentially because they are all patients with Tourette. So if you consider this level of complexity is not surprising, the lack or the difficulty at replicating findings in different samples when you're dealing with complex neurodevelopmental disorders. To further prove that genetics is useful, but is only the beginning of the stairway, the lowest level, the lowest step in the stairway, what is the informative content that copy number variants carry when they are searched in newborn siblings of autistic children? So newborn siblings of autistic children, you know, are at high risk of developing ASD. If they have causal CNVs, which by itself already is relatively rare because only 13 out of 157 siblings in this sample were found to have what could be pathogenic or likely pathogenic CNVs. If those siblings, newborn siblings, are then followed on later, you will find that 6 of them evolved toward ASD, 5 toward atypical development, meaning they may not develop ASD, but rather ADHD, language delay, some kind of language development disorder, or they may develop learning disabilities of some kind or other, but not ASD. And 2 will go toward the typical development. So by no means can we say that these genetic findings at birth are sufficient, at least if you're dealing with, if you're looking at CNV, so if you're looking at array CGH, are sufficient to really hold enough predictive value to be clinically valuable. And we have found even more discouraging results in our sample. So to make a long story short, genetics is useful if you find something wonderful, but what if you don't find anything, or if you find variants which are really by themselves not sufficient to explain what you're looking at clinically, the severity of the symptoms. And this is really almost the rule. I would say it's the most frequent outcome. Well, in order to understand what these small little black hyphens really mean, the common variants, I think it's useful to move to the second step of the stairway and to merge the information that we get from metabolomics with the information we have from the genomics. You'll know that epigenetics essentially means how tightly the DNA strand is wrapped up around histones in chromatin tertiary structure. DNA is so long that unless it was wrapped so tightly around histones and in such a wonderful architecture into chromosomes, there would be no way that DNA could really fit into such a small space as the nucleus. So it's very important that this orderly fashion, that this orderly structure be maintained, also because DNA has to produce ultimately the building blocks of our systems, of our organs. And in order to produce proteins, it must be transcribed into RNA. So the machinery, the protein machinery for transcription, which is really very complex machinery, needs to find enough room, enough space to reach its target regions on DNA and to start transcribing DNA into RNA. If DNA is wrapped around histones too tightly, there is simply no way that this transcriptional machinery can perform this task. And therefore, what happens is that you may have gene silencing due to this mechanism, which may not differ from the gene silencing you would see if you had entirely lost the gene because of a deletion. But this process is really clinically meaningful, has been proven over multiple times. For example, monozygotic twins are supposed to be 100% identical at the genetic level. Now, today we know that this is not entirely true, but it's vastly true. So for now, for our sake, we shall hold this we shall hold on to this point. Now, if two monozygotic twins are different, are discordant for an autism diagnosis, meaning that one is physically identical to the other, but one is autistic and the other is not, clearly it shouldn't be because they differ at the genetic level, we just said, but they do differ at the methylation level, meaning at the level of the changes that occur in DNA and consequently also in histone to determine whether the DNA will be more or less tightly wrapped around the histones. So epigenetic control, they do differ a lot more than concordant unaffected twins. So in other words, if you have two twins who are 100% identical and they are non-autistic, they will differ by methylation only in as much as the white, the clear gray columns, meaning about 1%. If they are discordant for autism, they will differ by as much as 6 or 7 or 8%. So this is a very significant difference in between monozygotic twins who are concordant or discordant for autism. And this study that we collaborated with Simon Gregory and Margaret Perkach-Vance a long time ago, but it's a very interesting study, I think, because This is one of our families. I was working in Rome at the time, and this is a multiplex family. You have 2 affected children, and we sent the DNA of this family to Simon, and he found essentially that one of the 2 affected siblings had a deletion that was essentially involving the entire oxytocin receptor gene. So oxytocin receptors are extremely important. We know that oxytocin plays a major role not only in lactation, myometrial contractions, of course, but also in social cognition. So this was an interesting result, but what was puzzling is that his brother did not have the deletion. And you have the same family with 2 autistic brothers. But what happens is that his brother had something that was really, really surprising, which is that if you go around this site, pre-transcription start site, I think it was the minus 924 and minus 901 CPG islands. These are sites in the DNA that can be methylated. This brother had essentially entirely, vastly methylated these 2 CPG islands, a lot more than the rest of the family. So in other words, what in one brother, P, was obtained by a deletion, loss of one allele, in the other brother was obtained not by a genetic deletion, but a gene silencing mediated by methylation and tightly wrapping up of the chromatin. And this mechanism may have been very important also in other pairs, because we looked at in our laboratory at postmortem brains. This is superior temporal sulcus, area 41, 42, Brodmann areas 41, 42, so primary auditory cortex, so to speak. And you can see that in that region, oxytocin receptor messenger RNA was clearly decreased in several pairs, not all of them, but in several pairs to a large extent, and none of these had deletions. So this decrease in oxytocin receptor expression was clearly mediated in the brain of these autistic individuals by epigenetic mechanisms. And epigenetic mechanisms are so relevant that you can actually identify a set of methylomic markers that can distinguish your sample of autistic individuals from a sample of unaffected siblings with 99.7% of accuracy. And some of these methylomic markers are located upstream of genes like neurexin-1 protease and kynurenine-3 monoxygenase, which are really important as neurodevelopmental genes, as autism genes, etc. So now you may say, wonderful, game over. We found the panel of biomarkers that we were looking for, 99.7% accuracy. Who would want more? Well, there is one major caveat, which is that methylation is tissue-specific, ethnic group specific, region specific, environmentally driven. So you can be sure that you can go in Modena, you can go in Washington, DC, and you can go in Japan, and you can be sure that this panel will by far not maintain the same accuracy, and if anything, may not at all distinguish autistic individuals from unaffected siblings. So what this saying is that a posteriori, afterwards, you can tell that this is what you need as a combination of markers to differentiate ASD from unaffected siblings, but a priori, there is no way to know that this is going to be replicable. But it just goes to show that there is clearly a different pattern in methylation of DNA sites in autistic individuals and unaffected siblings. Now, things become even more critical when we go and find, and we meaning Janine LaSalle and this wonderful set of researchers, when we find that this difference in methylation patterns is not something that you find only or exclusively in autistic individuals who have been diagnosed, who are 4 years old, 10 years old, etc. You find it in the cord blood on the day of birth, cord blood collected from children, newborn children, who later will be diagnosed with autism. So this epigenetic dysregulation is not produced by autism, consequent to autism or concomitant with the onset of autistic behaviors is present already at birth. And it's clearly present at birth. You can see here the scatter plots and, excuse me, and it's different in males from females. So in other words, the genes, and many of these are autism genes, by the way, the genes that are differentially methylated differ between autistic males and autistic females. And one thing that to me is even more concerning is that differences in methylation patterns are not only present in cord blood on the day of birth, but they're present in sperm cells of fathers of autistic individuals. Now, I don't know about you, but when I was in medical school, there was no much talk yet about DNA methylation. But what was said is essentially that when sperm cells fertilize, when sperm cell fertilizes the egg cell, the oocyte then undergoes rapid demethylation so that any environmental influence, any environmental modulation of methylation processes in DNA would have been totally cleared in the early embryo, and then the early embryo would be re-exposed to a new environment and to its story, its life, and that would have generated an entirely novel methylation pattern, different from parental methylation pattern. Now, this turns out to be mostly true, but not entirely true. There are methylation sites in sperm and egg cells that are maintained when the embryo then develops. And these methylation sites are sometimes upstream or located within genes that are important for neurodevelopment. And there are multiple examples in this study of neurodevelopmentally relevant genes like this NORD group of genes that, depending on how much they are methylated or not methylated, methylation in paternal sperm is correlated with AOSI scores, meaning that the offspring is more or less autistic in correlation with how much these genes are methylated in paternal sperm. And this is impressive, I think, because it's beginning to point toward the fact that when we deal with the pathogenesis of ASD, we have been mostly thinking of the child that is autistic, the child that I'm diagnosing today, the child that is in front of me, but perhaps we should have been thinking more of what happened 10, 20, 30 years ago to the parents, to today's parents, because maybe that's when something happened that influenced methylation patterns that are now being discovered, having consequences in the second generation. And we shall see more of these as we go on. Now, interesting to see that genetics has a role, interesting to see that DNA methylation has a role, and clearly methylation can indeed contribute to increase the expression of the penetrance of genetic predisposing factors, genetic predisposing variants. But there is an even more interesting piece of information when we go and look at the meaning of the common variants. Now, we said before, common variants are distributed throughout the population. You have them, I have them. Some of them will slightly increase the expression of some genes. Some of them will slightly decrease the expression of some genes. It depends on which allele you carry. Each one will contribute a small percentage of variance. Now, if we look at, these authors have looked at the function of these common variants, of these SNPs, single nucleotide polymorphisms, not only in terms of which gene they influence, but also what is the mechanism through which these common variants may be affecting transcription rates. And it turns out that significantly more than expected, these common variants are really methylation QTLs, meaning that they are CPG islands, essentially, whose methylation will influence the expression of critical genes important for multiple functions. And one of those functions is immunity, immune-related pathways. So now this is really interesting because this is telling us one thing, which is that if you have involvement of genetic mutation, deletions, and duplications in neuronal genes or in chromatin gene expression controlling genes, then that's one thing, but immunity can also contribute, perhaps downstream in some way, if you like that term, through the control of immune-related genes by these methylation mechanisms. And this was especially interesting to us because several years before, I had been in Karoli Mernick's lab in Pittsburgh to look at the genome-wide expression of postmortem brains. Again, we are here located in the primary auditory cortex, a region which is clearly very important in the functioning of autism brains. And what came out of that study is that the category of genes that is mostly overexpressed and most critical in differentiating the expression patterns, the transcription patterns of the brains of autistic individuals from those of controls are really immune genes. Immunity meaning innate immune response. Here you have the pathways, and acquired immune response. Here you have other pathways. And while this study was not enormous, we had 6 brains matched, 6 brains of autistic individuals matched with 6 brains of controls. You can see that out of those 6 brains of autistic individuals, only 1 did not obey this rule and was missed by this analysis and confounded as an unaffected individual. But essentially, this overexpression of immune genes was really one of the hallmarks of these brains. And if anything, there was a slight underexpression of neuronal genes, but less visible. This result was again replicated and extended by Dan Geschwin a few years later in the same region and also in the frontal cortex. And again, what they found is that neurodevelopmental genes tend to be underexpressed, whereas immune genes tend to be heavily overexpressed. So this finding is now replicated in multiple brains of autistic individuals, is present already in brains of children who are very small, 3-4 years old, and is present in multiple brain regions. A study that we've just published looked at transcription levels, and now, by the way, you have noticed that we have moved to the 3rd level. We've moved up from epigenetics to transcriptomics to mRNA sequencing studies. We've looked at using RNA-seq in collaboration with the University of Milan, San Raffaele University. We've looked at 31 pairs of autistic individuals. And what happens, to make a long story short, is that we have one module which is clearly underexpressed, and it is a module that once again brings us back to the same pathways. Again, it's cytosolic ribosome. It's really all about control of transcription, control of the transcription of DNA into RNA. This module includes 82 genes, and the hub gene is RAC1. Now, this was a very interesting finding because in some way it brings together the genetics that we've seen at the beginning of my talk with transcriptional control as being one of the pathways that, if genes are mutated, deleted, or duplicated, if there is a genetic issue, then you develop autism. Now we find the same pathway to be involved in the pathophysiology. And the interesting thing is that this module and RAC1, here you see the hub gene. RAC1 is this propeller-like looking protein that binds different proteins with each one of these domains. And one of the proteins that it binds the most is protein kinase C beta 1, which is a protein that we had looked at in 2009 that has a major role in controlling neuroinflammation, among other things. RAC1 regulates neural development, and RAC1 is strongly modulated by endocrine disruptors. Now, this pathway, this module, is really hence having to do with phenomena that are transcriptional regulation, environmental exposure to different environmental pollutants, and with neuroinflammation. Now, I'm not saying by any means that RAC1 is really the critical gene. I'm not making, again, the mistake of talking about one gene as being the autism gene. But what I'm saying, essentially, is that we are finding, even at the peripheral level, these are not brains, this is blood, but we are finding an orchestra that, again, points toward the same recurrent pathways, the same recurrent mechanisms, and which seems to be deranged to a statistically significant extent. And this is also in line with proteomic studies, which are a fourth level of analysis that I'm not going to be discussing with you, that both peripherally and postmortem has found essentially, primarily, inflammation, peripheral and central. So why should we have increased inflammation in autistic individuals? In order to try to respond to this question, I will have to make a short introduction to the concept of alternatives pricing, just because I'm not sure that everybody is familiar with this concept. I will try to briefly explain this concept using this graph. Now, imagine that you have a segment of DNA at the top, which has to encode RNA, which then has to be translated into a protein. And imagine that in the brain, we want to have protein A. Now, nature tends to apply strategies that will reduce its energy expenditure in its effort. So it will produce an RNA, a precursor that is in line 2 here. This is the precursor, but then this precursor will be cut into segments 1, 2, 3, 4, 5, which once translated will become the protein that you see down here as named protein A. Now, it may be that the same protein, imagine it's a second messenger, it's a channel, whatever you like. It's a protein kinase, it's a channel, or whatever you like. Imagine that the same protein must also be expressed in the salivary glands, but that you need a slightly different version, one that has the shape of protein B. What Mother Nature will do is use exon 1, 2, 4, and 5, cut out exon 3, jump exon 3, and it will produce protein B. Now, if this mechanism of alternative splicing of RNA to lead to the maturation of messenger RNA is deranged, you may come up both in brain and in salivary glands with protein C, whereby exon 1, 2, 3, 5 are put together, but in some regions I did not want exon 3, and in others I needed exon 4. And protein C is neither similar to protein A nor identical to protein B. And it may not work exactly the same by any means. So in this case, if you think about it, if you go and run a genetic study, DNA won't tell you anything because DNA is fine. What you will find is that it's downstream of DNA that things have not worked appropriately, and that the messenger RNA is not what it should be, and the protein is also not what it should be. Now, why am I describing you this phenomena in connection with the 4 steps that I've described so far in terms of biomarker research? Because we know that in the brains of autistic individuals, alternative splicing is frequently abnormal. So there are abnormal splicing isoforms that affect function. Why does this happen? Now, it may happen for multiple reasons. There are genetic mutations that occur in exon-intron junctions. There are multiple reasons why this could happen, but there is one that I think is extremely, extremely important at this time. And I came to evaluate, to assess this reason, this mechanism through the pathway of metabolomics. We looked at urinary metabolomics. This is one of the two studies that we've run. We've just published the second study last year in collaboration with the University of Viterbo, whereby metabolomics at the urinary level can distinguish autistic individuals and controls. But the point that I would like to make here is not so much that there are different metabolites, but that in all studies that you will see, all studies of urinary metabolomics, will find and will describe some compounds more present in autistic individuals compared to controls, compounds which are not of human origin. These are essentially from the microbiome. So the microbiome, the 1.5 kilogram body weight of gut bacteria that we are whole carrying around, essentially is extremely metabolically active, and it produces compounds that are sometimes quite unbalanced in autistic individuals compared to controls. Now, this is such a consistent finding that we find in study after study, that this is what brought my attention to then try to integrate the microbiome into this picture that goes from genetics to epigenetics to now transcription and protein levels with this inflammatory component. Now, again, we all know that autistic microbiomes are different from typical children. They have more inflammatory strains like Clostridia. And we also know from this wonderful, wonderful study by Masmanian and many other excellent, extraordinary groups. From the Cell study in 2019, we know that the microbiome of autistic individuals can have profound effects on the neurodevelopment. Let me describe to you this study for those of you who haven't had the fortune to look at it, because there are at least 2 points that I would like to make. The first one is, these are germ-free mice, so they have, their gut is not colonized yet by gut bacteria. These germ-free mice are colonized by the investigators with gut bacteria coming from autistic individuals and from typically developing individuals. So you have two groups of mice, those colonized by autistic microbiotes and those colonized by typically developing microbiotes. You take these mice and then at 7-8 weeks, you breed them. So you don't look at behavior in these mice, you breed them. And then they have F2, they have the offspring, second generation. And these are the mice that then you go and study at the behavioral level. And long and behold, these mice at the behavioral level do display autism-like behaviors. And at tissue analysis, they will show essentially an abnormal, a dysregulated expression that really speaks not so much to a difference in neurodevelopment, but rather as a difference in alternative splicing mechanisms. In other words, the microbiome is inducing abnormal splicing, abnormal alternative splicing, which involves at least 52 genes that are listed in the SFARI database as being autism genes. So going back one slide, these mice on the right, the second generation, the F2, have been exposed in utero to a microbiome present in parents, which was able to alter their alternative splicing in the brain, also in a fair percentage of genes which are involved in autistic disorder. Now, how does this ultimately then converge into what we see clinically in our children, in our patients, in our adolescents with autism. Now, we all know that autism is abnormal or altered behavior, which really stems from altered connectivity. This is, I think, fairly a common knowledge. Now, during the first decade of the year 2000, the focus was really mainly on disconnection, reduce connectivity and reduce functional connectivity between distant brain regions. So a reduced capacity to integrate information. And of course, when you have a problem with connecting distant brain regions and with parallel distributed processing of information, it is clear that social cognition will very much suffer from these, because social cognition does require a lot of convergent information shared by different brain regions. However, already at this time, it was beginning to be evident there were also some regions which were hyperconnected. But this idea that some regions may be hyperconnected came along a little bit later. I mean, we knew that essentially there was an excess in disconnection, in reduced connectivity. You see that all these samples collected in different American universities, or the sample of autistic individuals tend to have more blue compared to the controls to the left, more blue squares. So there is a reduced connectivity among many different brain regions. But essentially, this point was really not so much underscored until later, and more recently, much more recently, it has become very clear that when we look at connectivity in autism, there are really very different autistic individuals. Schematically, there is a subtype 1 that has very much reduced connectivity, which is what we were thinking all along between 2000 and 2012 or 13. But there is a subtype 2, which is really characterized by more hyperconnectivity. And many children, many patients really are somewhere in between, because they have partly hypo and partly hyper. And this generates an array of clinical pictures that is tremendous. And it's interesting to see how things make sense when you go to the genetics level, to the transcription level, because the regions that are hyperconnected are also the ones that exhibit greater excitation over inhibition gene expression. In other words, the Hebbian rule that whenever that neurons that fire together, wire together, remains even when connectivity is abnormally excited. So here you go, and you begin to try to merge all the information that I've tried to give you into something that connects directly with brain structure and function. When you look at functional connectivity, you can think of different brain regions, some of them being hyperconnected and some of them being underconnected, hypoconnected. And you can think of different subtypes of autistic individuals. Some of them have a predominant reduced connectivity, like subtype 3. Some of them will have a predominant and diffuse hyperconnectivity, subtype 1. Probably the majority will have a mixture of different pictures, and subtype 2 could be a good candidate, where you have increased connectivity that explains, for example, stereotypic behaviors or insistence on sameness, and reduced connectivity, which really explains a lot of difficulty in putting together sensory, multisensory information, and motor coordination. But nonetheless, the one thing that I find most interesting in this very recent approach to connectivity is really that one gene and one pathway. The gene is essentially PTEN, the pathway is mTOR. Let's focus on the pathway, which is connected with growth. is essentially overexpressed in those areas that are hyperconnected. And it is fascinating to notice that while this pathway, and by the way, PTEN here is one gene that when mutated produces autism spectrum behavior. Here you have two other genes that when mutated produce tumor-like syndromes, genetic syndromes, neurofibromatosis and tuberous sclerosis, which are also oftentimes associated with ASD. But the really interesting point, I think, here is that on the top right, neuroinflammation, immune activation can activate this pathway, and essentially the areas that have increased connectivity may be, this requires a little more work, but it seems like the areas with increased connectivity are also the ones that have more neuroinflammation. So, genetics, for example, with a PTEN mutation alone can produce autism spectrum disorder or can produce a very severe intellectual disability. Epigenetics can contribute, and you don't need to have such a mutation to have at the end an autistic phenotype. The gut microbiome may be also contributing prenatally, especially prenatally. We tend to work on the gut after birth when the child is already diagnosed, and that's fine. It may also contribute. But the experiments that I've shown you really move the timing of concern toward the prior generation, as does epigenetic exposure to different environmental factors. And all this converges into neuronal cells, which build networks. Those networks can be different in different brain regions based on differential expression patterns of the genes involved. And these may also reflect the differential level of neuroinflammation that is present in different regions. A degree of complexity, which is, to me, fascinating, extremely stimulating, and that is really telling us that altered brain connectivity, to conclude, can be due to genetic, epigenetic, gut microbiome, and neuroinflammatory components. And I could add on more, and I'm sure that many of you could add on more, but this is just to say that if we want to tackle complex disorders, we cannot and should not do what we were doing maybe a decade ago, which essentially was using biomarker research in a rather autistic fashion, each one working in their field of expertise. We need to put information together. And by putting information together, we are beginning to make sense of at least some pathways toward a pathophysiology. And those pathways toward the pathophysiology hold promise to give us strategies for intervention and for prevention that we would have never even imagined if we had remained linked to studying only a single level of this long stairway that I've shown you at the beginning. And I would like to thank again Steve and Paul for their kind invitation. And I would like to thank you very much for your attention. And I will be happy to answer questions. Thank you.
All right. Thank you so much for that presentation. People are typing questions into the Q&A section now. If you could leave your last slide up, that would be great. If you don't mind doing that, since we're recording. Thank you. So this first question, what mediates the phenotypic outcome, which is the end result diagnosis, if the gene set is pleiotropic?
The final outcome is really the final outcome is the same as white light. White light is not white. White light is the sum of red, orange, yellow, and whatever other colors. And it's not a sum of those, but it is the interactive conjunction of the seven colors in such a way that if you look at at their separation in a rainbow, you cannot take a pencil and write exactly the boundary between one color and the other. So what I'm saying is that the final phenotype that we see clinically is not the sum of the different levels, is really the core of complexity, which is the complex result of the interaction between all those levels. Plus, there is one last point that I would like to make, which is that the brain is a system made for survival and for adaptation. So it's the organ that more than any other is structured to interact with the environment. So having said that, the end result, we shouldn't be surprised that to this day, the diagnosis is really only clinical, because there is no single level alone, one single level alone, in my opinion, that will ever give you a diagnosis of ASD that is either sensitive or reliable. We will need to put together many different levels. And it may be that we won't be able to diagnose ASD, but we may be able to diagnose a subtype of ASD with different combinations of different biomarkers.
Well, this is a compliment for you. Absolutely brilliant presentation. You should have Dr. Persico every month. So somebody would like to have you every month. That's a very some kind words there. And then we did have one more question. This is from a parent perspective, because we have some parents here today. This is clearly such great research. How can people who are trying to learn more, parents, and do you have any suggestions about resources they can use to learn more about this in layman's terms as well? So obviously this is a high level, sophisticated presentation. Is there information out there that you're aware of that also helps people who are just starting and learning about genetics and autism to get on board and learn more about it?
I guess I'm thinking of some databases that are out there that may have information, like the Science Foundation, for example. I'm not sure that that's exactly what this person would be most interested in. I would have to think about it, because I think there are associations some parent associations that do provide more basic and layman information on this research. Right now, I cannot think of more than Safari, INSAR, and maybe a few more. I would have to think about it, actually.
Right, and we do have some of that information at ARI as well, some high level things on our website, but agree that it's very sophisticated. The Safari site does have a lot of great references, and some of those articles they do translate into layman's terms for parents. So that is a great resource. And we are running out of time, so Steve, I'm going to let you jump back in here. I think we've answered the questions that we've gotten.
Okay, well, I can't get the video going. But yeah, Tony, we appreciate all the presentation. It's going to be uploaded pretty soon to our website, which is also on YouTube. Actually, we have over a total of over 1 million views of our presentations from our webinars. So, but yeah, again, thanks. Let's keep in touch. And once we start asking for grant proposals, I'll definitely contact you, okay?
Thank you very much. Thank you for the opportunity of being with you tonight.
Oh, yeah, well, sure. Okay, keep in touch. Bye-bye.
Thank you. Bye.